video for cellular transport has to do with bulk transport. And in the previous videos to this one, we talked about how small molecules and water can enter or leave the cell through the lipid bilayer using transport proteins or just by simple diffusion. But large molecules can not cross the membrane usually in that way. And so they have to cross in bulk using those vesicles. And if you remember the vesicles, I like to call them like the cars of the cell. They have things in nice little packages and they move them either to other places within the cell or even out or into the cell between cells. But this type of transport also requires energy. There is one type of bulk transport called exocytosis. And if you remember, the root exo means out, kind of like exoskeleton. So in exocytosis, the transport vesicles are taking things out of the cell. And basically, this is anytime a product is being secreted from a cell, they'll use exocytosis. And so essentially what happens in exocytosis is the vesicles move to the membrane. So they're inside of the cell. They've got everything packaged up. They move into the membrane, fuse with it, and then release their contents out of the cell. And that's how they get rid of them. The opposite of that would be endocytosis, endo meaning into, and essentially it works in a very similar way, but in this case, the cell is taking in molecules into the cell. So, and this happens with moving molecules into the cell membrane by fusing vesicles with the cell membrane. There are 3 different types of endocytosis. So we like to call them phagocytosis, cellular eating, pinocytosis, cellular drinking, and then receptor-mediated endocytosis. So here you can see the 3 different types. We'll talk about each one individually. So phagocytosis is essentially when a cell engulfs another cell, basically by eating it. And how it works is they extend these things called pseudopodia. So if you know the root word pseudo, it means fake, and pod is a foot. So you guys see it's like fake feet stretching out, reaching around the food. And then basically what happens is it packages up into like a membranous sac, forming a food vacuole. And then that food vacuole is able to fuse with a lysosome and it can digest that food and be used in the cell. Pinocytosis, like the cellular drinking, is essentially when the cell gulps little droplets of fluid outside of the cell into tiny vesicles. And in doing that, it eventually gets full. And then what will happen is the vesicle will be taken into the cell. And then the vesicle can move to other parts of the cell. With pinocytosis, a lot of times the parts of the plasma membrane that form the vesicles get lined with this fuzzy layer of coat protein, so it kind of looks like little hairs. And these are sometimes called coated vesicles. And the reason for that is it helps them to move around the cell a little bit. So that's pinocytosis. And then finally, we have receptor-mediated endocytosis. So this is a specialized type of pinocytosis, so kind of similar to the last one. And this allows the cell to acquire bulk quantities of a very specific substance. So there may be receptors on the cell membrane that are going to receive particular molecules, and those are the main molecules that are going to be allowed to enter the cell. Those proteins have receptor sites that can bind to specific proteins or specific solutes. And then essentially those receptor proteins will receive the solutes. They're going to cluster in the coated pits, and then the coated pits will form a vesicle that can now move to the other parts of the cell. Notice that there are more of the receptor-mediated molecules, the ones that found their receptors, but there are still some other molecules that make it into the cell. And then basically lysosomes will digest what is in these vesicles and release those contents into the cell. So you can see here's, once again, phagocytosis. It's reaching out and grabbing this bacterium. The amoeba is eating the bacteria, and then it's putting it in its food vacuole. Pinocytosis, you can see those vesicles are starting to form with those coated pits. So these molecules right here are coming into the cell. Same here. And then finally, receptor-mediated endocytosis, you can kind of see the proteins here, and they're bringing stuff into the cell. And that's really all to bulk transport. Once again, it requires energy, and it's how we move a lot of large substances